Kowalski, and this past summer I had the privilege of working in Washington University, specifically Dr. Gautam Danforth's lab, to study horizontal gene transfer of antibiotic resistance genes in the human gut. So one of the main things that the Danforth's lab studies is antibiotics. And antibiotics are just molecules that target a certain area of the cell to kill. So one of the antibiotics I worked with was penicillin, which targets the cell wall, chloramphenicol and tetracycline, which targets protein synthesis, and trimethoprim, which targets DNA synthesis. But we know that not all antibiotics work because of antibiotic resistance genes. And antibiotic resistance genes enable the bacteria to grow in the presence of an antibiotic. And antibiotic resistance genes are becoming of increased interest in the health and science field because they're enabling pathogens in your body to grow in the presence of an antibiotic. So as you can see from this graph, the percentage of isolates is increasing over the time. Now, although this chart isn't really up to date, you can still see that the increase is going to keep rising. So the antibiotic resistance genes are making it harder to treat infectious diseases, which is causing a lot of problems in the health field, as I mentioned earlier. So one of the main ways that antibiotic resistance genes are passed along is through horizontal gene transfer, or HGT. And three main mechanisms that HGT can occur is through transformation, transduction, and conjugation. Transformation is when the recipient bacterium intakes DNA from its outside surroundings. Transduction is when foreign DNA is injected to the recipient bacterium from a foreign virus. And conjugation is when DNA is exchanged through direct contact between donor and recipient strain. So I worked with transformation and conjugation in my experiment. So we wanted to test what were the main driving factors of horizontal gene transfer and what antibiotic resistant genes were most commonly passed along. And we hypothesized that if an originally antibiotic-sensitive bacterium was resistant to an antibiotic, then horizontal gene transfer occurred and an antibiotic resistance gene was passed along. So in order to test this, we used various recipient and donor bacterium. So we used B. uniformis and E. fergusoni as the donor bacterium because they're commonly found in the human gut and they have known antibiotic resistance genes. And then we chose E. coli and Acinetobacter bailey, or ADP1, because they reproduce quickly and they readily take up foreign DNA. So first we started by testing conjugation, and we tested it between B. uniformis and E. coli. So first, we co-cultured the B. uniformis and E. coli in an anaerobic chamber for 5 days. And then we also added various antibiotics to this co-culture. So after 5 days, we took out the test tube, and we used the anaerobic chamber because B. uniformis is an obligate anaerobe, which means that it can only grow without oxygen. So when we took it out of the anaerobic chamber, we knew that B. uniformis would die, leaving only the recipient strain. So then we plated that out in hopes of sequencing the antibiotic resistance gene. And we found that after co-culturing for 5 days, B. uniformis and E. coli grew in the presence of chloramphenicol without any antibiotic in the co-culture. So this highly suggests horizontal gene transfer. And then we also found that E. coli grew in the presence of chloramphenicol when it was co-cultured with chloramphenicol. So these two colonies, or multiple colonies, indicates that adaptation occurred to the antibiotic chloramphenicol. So next we studied between E. fergusoni and E. coli. And E. fergusoni is a little different just because it's a facultative anaerobe, which means it can grow with or without oxygen. And in this case, we just used no anaerobic chamber just to make things a little easier on our part. So again, we co-cultured B. uniformis E. fergusoni and E. coli together for 5 days. We plated that out, and then since both E. fergusoni and E. coli would grow up together, we needed some way to select for E. coli. And then again, we would grow up the E. coli colonies in hopes of sequencing out the antibiotic resistance gene. So in order to select for the E. coli colonies, we used a fluorescent tagger called CFP. And you can see in the white light, colonies A, B, and C are all visible. So there's no really distinguishing factor between those colonies. But then when we looked at the colonies under 480 nanometer light, you can see colonies A and B and not C. So colonies A and B are E. coli colonies, while colony C is E. fergusoni, so we can just ignore that for now. So we found that after co-culturing for 5 days, E. coli grew in the presence of chloramphenicol without any antibiotic in the co-culture. And next we studied between transformation. So we looked at transformation between ADP1, which is again the Acinetobacter bailey, and pooled DNA. And one cool thing about the Washington University lab that I worked in, they were part of the 100 Genome Project, so I used some of the leftover DNA from the 100 Genome Project for the pooled DNA that I used. So I first started by plating out the ADP1, adding the DNA to it, letting that grow together, and then adding broth to slurry up the cells, and then plate that out in hopes of growing up the colonies and sequencing out the antibiotic resistance gene. So we found that when ADP1 was paired with the phylum or the class Bacteroidetes, it grew in the presence of chloramphenicol. So again, this highly suggests horizontal gene transfer occurred. So you probably noticed a trend with chloramphenicol, and so did we. We found that chloramphenicol readily transfers between the donor and recipient strain, and it transfers regardless of relatedness. For example, B. uniformis and E. coli are not very closely related, but E. fergusoni and E. coli are very closely related. So it was interesting to see that relationship between the two. And then eventually, because the chloramphenicol antibiotic resistance gene is so readily transferred, chloramphenicol antibiotics could become ineffective when treating an infectious disease in your body or something. So antibiotics that have the chloramphenicol antibiotic in it should be more regulated in the health and science field to prevent this from becoming a bigger issue than it already is. But our experiment isn't necessarily done just because we wish to sequence out the antibiotic resistance gene that we found, or so that we think we found. And then we would also like to test other donor strains. We originally wanted to test 4 or 5 more, but due to contamination, we were unable to do this. So that would be another step that we could take in this process. And this experiment would not have been possible without the entire Davis lab, especially Molly Gibson and Vitas Wagner, and then also the Young Scientist Program who let me have this opportunity. So thank you.